Hello everyone, welcome to the final lecture for the 2020 Spring Intro Biochem course. Okay, so today we have two, maybe three learning objectives. First, we're going to compare and contrast DNA and RNA, and sort of as a sub-objective of that, we're going to compare and contrast the stability of DNA and RNA. If you look at your handout, you should be able to see a compare and contrast box right there on the first page. You can use that if you like to list the similarities and differences between DNA and RNA. Next, we're going to describe the three steps or events that occur in DNA transcription. That's the process by which your DNA gets read and transcribed into RNA, which then later on the line gets converted into protein. Alright, DNA and RNA, nucleic acids. So they have similar structures and similar names. So DNA and RNA are both polynucleotides that are linked by phosphodiester bonds. We'll look a little bit closer at the structure here in a minute. And in this structure, the 3' carbon of the first subunit is connected to the 5' hydroxy of the second unit. DNA has 4 bases, A, C, G, and T, whereas RNA has A, C, G, and U. So in DNA, where you see thymine, in RNA, that's going to be uracil. So for DNA, the sugar is deoxyribose. For RNA, the sugar is ribose. So that's where we get RNA and DNA from. DNA, deoxyribonucleic acid. RNA, ribonucleic acid. So deoxy means that it's lacking one hydroxy group. We'll get into that a little bit later, but that has enormous implications down the line. So let's look a little bit closer at the structure. So anywhere here where it says base, that's going to be the thymine, adenine, those structures that you memorized from the previous lecture. And this is the sugar phosphate backbone. Here's the sugar, deoxyribose for DNA, ribose for RNA, and here's the phosphate backbone. One sugar unit linked to phosphate, linked to another sugar unit linked to phosphate, and so on. And on the side of each of these sugar subunits is the DNA base. That determines the identity of your DNA sequence. And here's what I mean when I say 3' and 5'. So if we start counting here, this is carbon number 1. 1, 2, 3, 4, 5. 5'. So the 5' carbon, say right here, is attached through the phosphate backbone to the 3' hydroxy on another sugar. So here would be a 5' linked through the backbone to a 3'. Or I guess if we're looking at it the other way, 3' linked through the backbone to 5'. Same for RNA, exactly the same, with only two differences. One we already mentioned, instead of thymine we have uracil. And also we've got this extra hydroxy right here. Remember DNA deoxy, it's missing that hydroxy group. RNA, not deoxy, it retains that hydroxy group right here on the 2' position. Okay, so DNA in your cells exists as a 2-stranded double helix. So we already know about the alternating sugar phosphate backbone. So you take 2 of those strands and they're joined together by non-covalent forces. That force is hydrogen bonding. Blast from the past. Lecture number 2, I think. So RNA only has 1 strand, so it's not stabilized by this hydrogen bonding that exists in the double helix. And each rung on the double helix in DNA has a pyrimidine and a purine. And hydrogen bonding occurs between these bases. The sugar phosphate backbone makes up the backbone, but the actual bases, A, T, G, and C, participate in the hydrogen bonding. So A bonds with T, and G bonds with C. So if you hear that these base pairs have complementarity, this is what it means. A on one strand will always match up with a T on its complementary DNA strand, whereas G will always match up with C on its complementary strand. So one strand of DNA will have an entirely sort of a mirrored strand on the other one, and these two hydrogen bond together to make a double helix. Let's take a look. So here's sort of a chemical structure of the double helix. Here on the sides, we've got the sugar phosphate backbone. The arrow indicates a 3' to 5' direction. 3' into 5'. Same over here, 3' into 5'. But in the middle, between these two sugar phosphate backbone strands, are the bases that are participating in hydrogen bonding. So T with A, C with G. So you can see that anytime you have sort of a hydrogen that matches up in between and a heteroatom, oxygen or nitrogen, that can participate in hydrogen bonding. So you can see A and T, they have 2 hydrogen bonds in between them. Whereas G and C, they have 3 hydrogen bonds. So the exact orientation and number of these hydrogen bonds means that the DNA can be read as a specific code, if you will. Since a C will always match up with the G and an A will always match up with the T, it's a unique way to save data, basically. Additionally, this provides some of the stability of DNA. These are sort of holding the 2 strands together. They're one of the forces that hold them together. So these base pairs stack on top of each other. On the right, you can see sort of the stereotypical double helix cartoon. So the sugar phosphate backbone forms the sides of this ladder or spiral staircase, or whatever you like to think of it as. But that doesn't necessarily make it a helix. One of the reasons that it's a helix is the interactions of these AT and GC base pairs. Now, we already talked about the hydrogen bonding, which helps hold these together. But there's also these stacking interactions. So this is sort of a subtle point. Something actually you don't learn until a lot later in chemistry exactly why this occurs. But I'll see if I can explain it to you guys here. So these base pairs, since they have these planar structures, if you stack these on top of each other, the electrons have attractive forces between adjacent base pairs. And I don't mean adjacent base pairs on opposite strands, I mean within the same strand. So if you stack a lot of these up and twist them in a double helix, it really stabilizes that structure. Number 1, through the hydrogen bonding between the strands, and number 2, through the stacking interactions within each strand. So 2 major ways to really stabilize DNA. So if these are really tightly wound up in a double helix, if you want to ever use your DNA for something, if you want to read it to see what the genetic code says, you're going to have to unwind it. So applications would be like DNA replication. If you want to make a copy of your DNA, well, you need to open it up, you need to unzip it, and read what it says, and make a copy of that. Alternatively, much more commonly, if you want to make an RNA transcription to eventually make some protein, well, you're going to need to open up that DNA double helix and read the template DNA strand to see what it says. Sort of like a book. You can't read a book until you open it up. You've got to unwind the book, so to speak. So in order to unwind DNA, you actually have to break these hydrogen bonds. I guess your enzymes actually have to break the hydrogen bonds. You're not really thinking about it. So as we know, AT base pairs only have 2 hydrogen bonds per base pair, whereas a GC base pair has 3. So AT base pairs are a little bit more, it's easier to open them up, easier to unzip those hydrogen bonds than it is for GC base pairs. Because of this reason, a DNA strand with too many GCs in it will not be, I don't want to say, it'll be biologically troublesome. And if you want to learn more about DNA, I suppose in like a future class, or if you're just curious, that's sort of an interesting thing to look at, the AT to GC ratio. Okay, so that's a lot about DNA. DNA is super stable. It's got those two forces helping to stabilize it. RNA, on the other hand, has one force that's really, or two forces that are really trying to chew it up. It's really unstable. So first, RNA is inherently somewhat unstable due to that 2' hydroxy group. Remember, DNA is deoxy. It doesn't have that hydroxy group, but RNA does. And the great thing about that hydroxy group is it makes it really easy to hydrolyze RNA. It makes it really easy for your RNA to fall apart. But maybe that's not enough for your body. Maybe your body wants to chew up RNA even faster. So your body and other cells and the entire world are covered in these things called RNases, which actively chew up RNA. So not only is RNA inherently kind of unstable, it's easy to hydrolyze, there are a lot of enzymes that chew up RNA. And one of the main reasons for that is because viruses can be made of RNA. So if you can chew up RNA, you can prevent viruses from infecting you. So because of these two things, the half-life of DNA, the time it takes for DNA to fall apart, or half of DNA to fall apart, is about 500 years. Whereas for RNA, that's about 5 minutes. So back when I was about in college, actually, I did a summer internship at the Noble Foundation down in Ardmore, Oklahoma. It was a great time. If you get an opportunity to work down there, I highly recommend it. So I was there for almost 2 months, and I was doing research on RNA. So we grew some test plants, and we had 2 different groups of plants. And then at the appropriate time, we ground them up and extracted the RNA from their cells. And it took about a week to do that work. It had a lot of steps in it. And at the end of the day, once I got the RNA out of all of my different plants, we looked at it and it said, oh, your RNA fell apart somewhere along the way. So in order to keep RNA from falling apart, you have to keep it really cold so hydrolysis is slow, and so the enzymes that chew it up can't proceed. And when I say really cold, I don't mean freezer levels. I mean like liquid nitrogen levels. So somewhere along the way, I made a mistake and left it out of the liquid nitrogen too long, and it all fell apart. A similar story from when I was in grad school, I was working with some DNA, and I put all my samples away, but I forgot one on my bench, and I didn't use that one for a long time, and I actually didn't put that DNA back in the fridge for an entire month. But it was completely okay. DNA is really stable. Everything loves DNA, whereas RNA is not long for this world. So think back to Jurassic Park if you have trouble remembering this. Was it dinosaur RNA inside the amber? No, it was dinosaur DNA. I mean, as we know, dinosaur DNA still couldn't last, millions of years. But it can last 500 years, whereas RNA, as soon as you take it out of the freezer, it's falling apart on you. So we already discussed some of the reasons for that. But basically, DNA is the unalterable code in your cells, whereas the RNA is the disposable sticky notes that your cell uses to send messages, get things done, but it doesn't want them around forever. Great. So let's talk about transcription. So this is the process by which your body reads DNA and transcribes it into RNA and eventually into protein. So there's 3 types of RNA that are required in transcription. First, messenger RNA. So this is the template for protein synthesis. Second, ribosomal RNA, or rRNA. So this forms a little over 50% of your ribosomes, and ribosomes are the protein synthesis machinery inside your cells. These are the factories that make proteins based on the instructions that are encoded in the mRNA. The rRNA they form a physical factory. Finally, there's transfer RNA, tRNA. So these carry free amino acids around, and they match up with the template, your mRNA, messenger RNA, to assemble these amino acids in the correct sequence. So they carry amino acids, and they recognize where they're supposed to go. So as we already mentioned, in order to transcribe DNA, we have to open the DNA duplex. We have to unzip the double helix. So this makes the single-stranded DNA template. And polymerases that make the RNA, so RNA polymerases, act in a 5' to a 3' direction. So that's shown on this figure right here, with the big arrow pointing to the 5' and all the way down to the green arrow pointing to the 3' on the bottom. So if you're in a future class and you're studying this more in depth, and you hear people talking about 5' to 3', 3' to 5', what they're talking about is the direction in which the DNA sequence is read. So if you're making RNA, it's read from a 5' to a 3' direction. So we'll start off by looking at RNA transcription in prokaryotes, say E. coli or some other bacteria. This is going to be a little bit different from how eukaryotes like us perform RNA transcription. So only one strand of DNA is the template, and RNA is going to bind somewhere on that template strand of DNA upstream of the start site. So the reason it binds upstream of the start site and not at the start site is because it needs to prepare. It needs to bind correctly and orient itself in the correct way before it starts transcribing the DNA into mRNA. So in order to tell the polymerase where to bind, there are these specific sequences called promoters. They promote binding, and they're required. So there are various different ones. One that's most commonly known is the TATA box. So that's shown here in this small blue box here. And there are various, promoters, and there are various TATA boxes. And they're a binding site for RNA polymerase. So RNA polymerase is going to bind there, prepare to transcribe DNA, and then once it goes downstream, it'll chug along and, well, make your mRNA. So yeah, still looking at the bacterium E. coli as sort of a model organism, there are 3 basic events that occur in transcription, 3 steps, if you will: initiation, elongation, and termination. Let's walk through these. So initiation. RNA polymerase needs to bind upstream of the start site. So it'll bind to a promoter sequence, such as the TATA box, or a Pribnow box, or some other conserved sequence, a sequence that's the same across all of those E. colis. So once RNA polymerase binds, it'll bind weakly until it finds the correct promoter, and then once it finds a promoter, it'll bind tightly and prepare to transcribe. So this polymerase consists of multiple subunits. And so these work together, and they open up the DNA double helix once they recognize that promoter, and they'll synthesize that short segment of RNA, that mRNA sequence. Great, so it's initiated. Once it started transcription, well, it's going to elongate that transcription. It's going to keep going. So one of the subunits, the sigma subunit, is going to dissociate from the RNA polymerase. And this basically unlocks the polymerase and allows RNA transcription to occur. So, well, it'll occur. And the polymerase will sort of chug along until it terminates. So the third step, termination. So once it hits a termination sequence, a signal that tells it, this is the end of my mRNA. It's time for me to stop. Well, it'll stop. So RNA polymerase will dissociate from the DNA, and the transcript is complete. For some of these, a protein called the Rho protein may become involved. So this disassembles the RNA polymerase once it's completed its job using ATPase activities, phosphorylating it, similar to how we've seen other proteins work. So those are the basic 3 steps: initiation, elongation, and termination. So how about transcription in eukaryotes? So it's a little bit similar for eukaryotes like us, but we've got some different tricks that prokaryotes don't have. You can basically think of us as sort of like the high-tech organisms on this planet. So we have some specializations. So for example, eukaryotes have organelles. Transcription for us occurs in the nucleus of cells, and protein synthesis occurs elsewhere within the cell on another organelle called the endoplasmic reticulum. Bacteria don't get to use these advantages, so good for us. We also have some interesting processing reactions. So if you remember, well, from about 5 minutes ago, I was telling you that RNA is very unstable and it lasts maybe several minutes if you're lucky. So we have different ways to stabilize these for a little bit longer so that our body can use them for a little bit longer. So one of these is a 5' cap structure. So this is a 7-methylguanylate cap, a picture of which is shown here. So what you do is you stick this to the end of your RNA, and this does two things. It orients this mRNA correctly for protein translation, which occurs with ribosomes, and it protects it from degradation. So if you're going to hydrolyze this, well, you have to start from the end. And if you've got a cap on the end, that's really going to slow it down. But that's not the only trick we've got. We've also got something for the other end of mRNA, the 3' end of mRNA. So another enzyme called poly-A polymerase, well, it sort of does what its name says. It adds up to 250 adenine residues, so poly-A, onto the end of mRNA. So this makes something called a poly-A tail. So this process is called polyadenylation. And it does sort of the same two things as the 5' cap. So this first helps orient mRNA for correct protein translation, and it also protects RNA molecules from degradation. So you might think, well, this poly-A tail sounds like you're just dumping a lot of trash on the end to this mRNA that I worked really hard to transcribe. And that's sort of exactly what's going on. So if you have hydrolysis occurring, or chewing up this mRNA from the end, if you stick 250 letters of trash onto the end, you've got a good buffer space. So until those 250 have been chopped off, you don't chop that important coding part of mRNA, so to speak. So you've got a cap on one end and this buffer tail on the other end. So that helps stabilize RNA within eukaryotic cells. So another thing that we do is called splicing. Once again, like all of the topics covered today, you can get a lot deeper into these if you wish, say in a future course or just by your own curiosity. But basically what happens is DNA is formed of exons and introns. So exons are segments that stay in the final transcript, whereas introns are removed. So first, the entire thing is transcribed, and then it's processed to remove any extra introns that may be extant. So the final mRNA is only consisting of exons. Okay, so that was our sort of a 20-minute dive into DNA and RNA. It's our final lecture of the semester. Let's summarize it really quick. First, DNA and RNA are really similar in structure, but subtle differences allow them to play separate roles within cells. And a lot of that subtle difference is entirely on that loss of that 2' hydroxy group from RNA to DNA. So DNA is incredibly stable. It can last 500 years. Like in my story, if you leave it on your countertop, it'll be okay a month later. Whereas RNA is incredibly unstable. It doesn't want to survive for too long at all. So like in my story, I took it out of the liquid nitrogen for just a few minutes somewhere. It was already gone. Next, DNA transcription occurs in 3 steps: initiation, elongation, and termination. So this is the process by which DNA gets transcribed into mRNA. And later on, the mRNA participates in a process to translate it into the protein final product. All right, well, I hope you enjoyed this lecture, and let me know if you have any questions.